Hi, I'm Preeti Bais. I'm from the Jackson Labs Connecticut branch. Today I'm going to talk to you about the MMap mouse metabolomics analysis platform. And to get hired at Jackson Labs was to promote metabolomics amongst the Jackson Lab researchers. And if I can convince one of you in this room to start looking into metabolomics, then I have done my job. So here we go. So the outline of my talk today is I'm going to introduce what is metabolomics. It's a very short introduction of metabolomics. And then what are the problems that we are facing as metabolomics researchers. And then finally I'll show you our solution, which is the MMap database and the pipeline. So here we go. What is metabolomics? This slide is a detailed version of the slide that Dr. Anton Kumar was showing yesterday. So he stopped at the protein, but I take it further and I show from DNA to RNA to protein to metabolite. You have to do everything or some of the things in combination to get a full picture of a cell's mechanism. So people have been doing DNA for a long time. Then RNA came, people started working on RNA also. Then they started combining DNA and RNA information, call it EQTL analysis, then protein PQTL. So the idea with metabolites is MQTL analysis. So when you do DNA and metabolite analysis, you get a more complete picture of the mechanism of the cell. And metabolomics is, or metabolites are closer to the phenotype. So they take into effect the genomic and environmental effects together to show you the mechanism of a living being, okay, or a cell. So a typical metabolomics workflow starts with like this. So you have two types of samples that you want to compare. And I purposely put two colored mice to show that they are different, but in normal life you won't see, you wouldn't want to compare these kind of mice. So the phenotype may not be as clear as I'm showing here. Then what you do is you take some sample. I'm showing blood, but it can be tissue or plasma or anything. And then you pass the sample through a LCMS or GCMS machine. What GCMS or LCMS machine do, they work like a prism where you put the sample in and they tell you the components of that sample. So if you put blood, they will tell you the metabolic components of that sample. And you recognize each component by a specific fingerprint, which is called the chromatogram. And finally, you have this kind of data, metabolomics data, where you have for each sample, so I'm showing mutant versus wild type, and you have different metabolites and the concentration, relative concentration of those metabolites. So as you can see in this picture, some of the metabolites in a biological system may be in higher quantities, and some may be in lower quantities, and some may be changing between the two conditions, and some may not be changing. So idea for a metabolomics analysis is to find out the metabolites that are changing between the two conditions that you are testing, and from that information, try to understand the mechanism of the biological system. So I started working at an R21 grant, and the goals for that grant were to identify metabolic pathways related to pathogenesis in mouse models of 3 neuromuscular degeneration mutants. So we wanted to start with 3 mutants and actually build the bioinformatics tools to start using metabolomics within JAX. So the idea was to generate the data for those 3 mutants, do the data analysis and interpretation, and while doing it, also create the software system that other researchers at JAX can use to start using metabolomics in their research. So I'm going to focus today's talk on showing you that software. So bioinformatics infrastructure is lacking in metabolomics because metabolomics is one of the newest omics technologies. So there are not as many researchers who are working in metabolomics. And we are facing different kinds of problems, like biochemists have typically been using different synonyms for the same compound. For example, B-alanine or beta-alanine are the same things. And there has to be a database which can find out like that B-alanine and beta-alanine are the same things. So otherwise, we cannot compare your data with somebody else's data. And then there are very few metabolomic data analysis pipelines that are designed for the biologists. So the initial inertia is pretty high for the biologists to start using a new technology. So we wanted to create the bioinformatics infrastructure so the biologists can feel that they can just do plug and play and actually see some results and before they talk to a statistician or a bioinformaticist. So some examples of metabolomics data pipelines are HMDB, which is based out of Canada, and then plantmetabolomics.org for the plants. This was actually my PhD thesis. So what we did in this plant metabolomics was we took 140 Arabidopsis mutants and created the database and the pipeline. So I'm trying to do the same thing for the mice for Jackson Labs here. And then the ultimate goal of a metabolomics pipeline is to combine the metabolomics data with the biological annotation and the other types of data that is out there, for example, genomics data. So here is the pipeline. So what we do is we take the samples that I was showing you in the previous slides, run it through the mass spectrometry, get the data out, and we have a mass spectrometer at our Bar Harbor campus. And then I created this metabolomics database, which follows the metabolomics data reporting standards. So these standards are very much like Miami standards for microarray data. They were called MyAmet standards, but now the name has changed to MSI standards. So what is the minimum information that we have to put in data reporting for metabolomics is listed there. So this database actually follows that. So once you use this database, you can actually publish your results easily. And then I created this data mining pipeline. So what this data mining pipeline does is it creates publication quality graphs with a click of a button, so you don't have to actually write any scripts or anything. And I'll go over in details and show you how it works. Okay. So MMap Features is an advanced data analysis tool. It's a menu-driven automated data analysis pipelines. It uses best practices in metabolomics data analysis. So one of the reasons for the inertia is for people to start using a new technology is that they don't know what are the best practices and if their data analysis technique is going to work in metabolomics or not. So this MMap database actually uses all the best practices in metabolomics data analysis through extensive literature search. And it's connected to genomics information because the ultimate goal is you want to find which genes are involved and which pathways are involved. So all that information is connected. You can do interactive plots. This is one of the biggest features of MMap data analysis pipeline. You can click on any graph. If you find some metabolite interesting, you can click on it and see all the details about that metabolite. It's a flexible pipeline, so you don't have to follow all the steps. You can stop at any step, download all the results till that step, take it to a statistician, and do your own analysis. So you don't have to stick with this pipeline till the end. And then there's also extensive help in interpretation of plots with examples. So whenever you click on any question mark sign, you can actually see a worked out example of how to interpret that plot. So these are some of the plots that it generates. And then help in interpreting the plot. So you can actually print the plot, you can download the plot, you can make it bigger, smaller, search for any gene, search for anything like in here. And then when you click on one of these icons, you can actually see the worked out example, which shows you how to interpret that plot. So when you see this kind of plots, don't get overwhelmed. You can just click on this link and see how to interpret that. Okay? And then interactive plots. So I'm showing an example of a plot here, and the user just used genotype and gave a threshold, which is also menu driven, and then it tells you what are the interesting metabolites at that threshold. So when you click on any of these points, you can actually see all the details of that metabolite. So what are the synonyms, what is the molecular weight, what is the chemical formula, and all those things. And plus, it is also connected to the genomic information through the pathways. So for example, this metabolite is participating in this pathway as a product. And when you click on that link, you can actually see that pathway. And you can download all the information at any point of time. Okay? And then we have also connected this information with the mouse genome informatics from Jackson Labs. So you can actually get all the MouseCyc information and MouseMine information, and they have been very well established projects within JAX, and they are available to general public as well. So here I am showing an example of the connection, MGI connection, and what you can see. So you can see the gene list in a particular pathway that you are interested in, and then term enrichment analysis, OMIM disease phenotype, and stuff like that. So all this information is available to a biologist or a user through just clicks of button clicks. So in summary, MMap provides a computational metabolomics platform for JAX researchers to incorporate metabolomics data in their research. And it will be available to the community as well. So we were debating whether we should release it now or later, but then we found out that Apple is going to introduce their Apple phone 6, so we kind of held back. That's just an inside joke. And then the automated pipeline is modular and can easily be extended to experiments to include different factors. So for example, I was showing you the mutation factor, but you can do any kind of factors in here, like drug, disease, or anything. So it's pretty modular that way. And then currently the genomic annotation for mice are available because it is built for Jackson Labs, and we are more interested in mice. Metabolomics in this particular project because we were using 3 mice mutants, but it can easily be extended to other organisms which have the biopsych pathways. For example, worm, fly, and yeast. And finally, this is the group. So we have 3 PIs. Dr. Cox, Dr. Burgess, and Dr. Bult are the 3 PIs. Lauren is an intern, and finally I'm there. And then it's funded through an R21 Metabolomics grant. And I also want to acknowledge that our Chief Scientific Officer, Dr. Charles Lee, is in the room. He is a world-renowned scientist for his discovery of copy number variations. So I really want to thank him for being here for my presentation. Thank you. Yep.
What would be the input of this pipeline? What will be the input of this pipeline? Will it be mass spec output, or is a list of the metabolites?
It can be, so the input is the mass spectrometry output, which is very much like what I showed here in this, okay? But this is just a sheet of like data. You want to interpret this data, right?
Yes.
So the output of the pipeline is the interpretation of that data.
So basically the input is mass spec data output.
Yeah.
So to use it, we have to use mass spec.
Yes. For now, yes. So the different technologies in metabolomics are NMR and mass spec. So this is focused on the mass spec, but the idea is that we can extend it to the NMR data also. So any data which looks like metabolite name and concentration level, you can easily use it, extend it to that. But as of now, it just uses mass spectrometry
because
data.
you perform several analyses, including PCA and forest, random forest.
Yeah.
So what is the pipeline like? Do you compare many mass spec data and produce some tree-like output, or do you just use just one mass spec output?
No, so you take one mass spec output, and then you can put it in this pipeline and see like all these graphs for that mass spec, or you can combine that also. Like, for example, if you are using, like, if you tested like brown mice with like black mice in 3 projects, you could easily combine those results as well.
My last question, short question is, when do you think you will release this pipeline?
It's pretty soon, yeah. So it's available for the internal Jackson researchers as of now, but we are hoping that by the end of this year, it should be available to the community as well. Yep.
I have a very quick question. Simple. Embryo data, placental data, is that also included?
It can be, yeah. So as of now, it can take any data which looks like this, where you have ID, sample ID, and some numbers and metabolite names or metabolite IDs. So it can come from any sample. Yeah.
I have a question about this. Since we needed the input data from the mass spectrometry, so what is in your database? Can we use your database without our data, I guess?
Yeah. So right now, it contains data from one of the 3 mutants that I was talking about. So it has one of the mutants here. The other 2 mutants, we are still working on the mice. And then once we have that data, we'll put that in as well. So once the site becomes available to the community, you can definitely look in, like you can see those
so
that
it's,
data.
So basically, it's only in the neuromuscular, kind
As of now, yes.
So everything we put in had to compare with that.
No.
No?
So every project is different. So you can actually, I'm not showing it here, but you can actually, before this, you can actually search for data. And suppose search for a gene, search for a mutant, and then you get into that. And then there you actually put what you want to compare. So you can compare, maybe I'm showing it in the next slide. Over here. So here they are comparing genotype, but there was mouse sex also, so you could compare the sex. And we didn't see much difference in male and female mice, so that's why I don't have it here. But whatever factors, experimental factors that the researcher put in, the database can take that.
What is your publication program plan?
Yeah. So we are writing a manuscript on this, and the plan is to publish that, to send it for publication by the end of this year or by early next year. And this is one of the first launches of this project.
Do you have a journal in mind, or some do you know?
So we are debating whether we should put it as a bioinformatics paper or a neuromuscular degeneration paper. So that's what we are thinking about right now. Good? Thank you.